Bianca, is there a way to send samples for research if we are not there this weekend?
She's going to come join our panel.
Okay. Yes, absolutely. So maybe you guys can put in the Slido, the email for the natural history study in the biobank, but we are absolutely can help collect samples remotely. So we can send you tubes in the mail and we'll send it with a prepaid FedEx bag to send it back to us. The tubes do expire, so we try to send them when we know you're going to actually go and get labs drawn. But if you're going to get labs drawn for another reason, and the phlebotomist will draw these extra tubes for you, that's a great opportunity to do that. So we work really hard with you to help coordinate getting those tubes to you and coordinating it with when it works for you and your family. We do a lot of that.
It's a really complex question, although it's going to come out very simply. Can you explain the correlation of the science to finding treatments?
Yeah, if you don't understand the science, then it's really hard to find a targeted treatment. You don't want something that is going to have a lot of off-target effects. And the way you find really specific treatments is by understanding the science in great detail.
And is there a risk to starting a treatment too soon before we know what the science is?
Absolutely. I think that if you don't know what the science is, then it is very possible to actually accidentally make the opposite effect with your treatment than you intend to have. So it really is important to know the science, test the therapeutic candidates. There are many different ways to do that. Make sure that all of those I's are dotted and T's are crossed. And I think it's also best practice to ensure that those experiments have been done in more than one lab so that you can have confidence that the data is replicable. And that will give you a lot of confidence that you're really making a change or a therapeutic intervention that will be effective and safe.
Can I add to? I think one of the challenges, which Stephanie did an amazing job going through all of this, is that that process of turning those genes on and off happens at different times in development. And one of the things that's challenging about treating neurodevelopmental conditions in general is that we miss our biggest window, right? Which is during the pregnancy when the brain is developing initially. And so it can also be a challenge, even if we understand the mechanism of how these genes are working, we may not have access to the right windows in time. So that's one of the challenges as well, in that the function of these genes changes based on what our body needs at different stages of development. And we had a good question yesterday about CRISPR. And one of the challenges with CRISPR is the same thing, is that these changes in ASXL genes were present at conception all through initial neurodevelopment. And that's not a time window that we generally have access to. So another challenge that makes it much more complicated and make sure that we want to use treatments that could potentially be beneficial and not harmful.
And that leads to another question here, which is, since our kids' bodies have already developed and have passed that window, are we talking about treatments that are going to be useful only for future generations, or is there hope as we develop treatments with this basic science that they will also be able to help the kids who are here today?
I think that Bianca can give a follow-up as well, but I think that because everyone will be in a different stage of development, then it's likely that because every organ has developed different kinds of cells that are functioning in a different way, that it's possible that the therapies may need to be specific for clinical symptoms in the brain or in the gut or in the liver or whatever the particular therapy may be. So that's also going to make it complicated. And we don't know that something that has been tested and shown to be efficacious in a neuron will work the same in a different organ system. So I think that development is an issue, but also organ system is an issue.
We still grow and change a lot after we're born, right? So I still learn things this week, a lot of things this week. So even though we know there's this phase that we kind of are missing out on during early neurodevelopment, we also know that babies are born with pretty immature brains and pretty immature bodies. They don't do very much, and there's a long process of getting them to adulthood, right? Any parent in the room knows that. So I think it's not that the window isn't there. The window is definitely there. And I think if there's a treatment that was safe and effective, we still have windows of treatment throughout somebody's lifespan with variable efficacy, depending on what you're targeting and sort of the stage of development of that individual. But our cells are constantly turning over. We're constantly changing. So I think it doesn't mean that the treatments aren't going to be available for children now. But there's a lot of movement in genetics too towards early identification, newborn screening. How do we identify individuals who have access to a treatment early so that you can start those interventions early? And so I think that's something the community will need to deal with as time moves on. And when we have a treatment, then there'll be a big push too to try and help make sure we're not missing kids really early if that's the window of opportunity we need to have the most benefit from treatment.
I'm going to toss it over to Amanda. How can the ARRE Foundation help with family fundraising efforts?
Ooh, that's a great question. So the very easiest sort of entry point is online fundraising. Facebook fundraisers are easy. You can set them up with just a couple clicks. We have lots of families in the room who have done that in the past. We also have families who have done local events. Laura and I, we throw a winter ball in Maine where we both live. It's a community event. I think we raised $6,000 this year, $5,000 last year. So there's lots of creative ways to think about how to engage your community. I'm looking at Melissa over here who did a motorcycle rally with her family, and they raised $4,000, $5,000, I think.
Yeah, I forgot. I think it's like 5.
Yeah. And so I encourage everybody to think about how can you engage your community, your school community, your church community, any part of your community that is interested in supporting you, and asking for their support and getting involved in supporting you and supporting our larger ASXL family. And we're here to help you with that. So my email is amanda@arrefoundation.org. If you have ideas, I would love to talk to you about them. We're also always looking for sort of bigger connections too. Family foundations, corporate giving. A lot of big corporate employers will match donations. So Laura, for example, works for American Express. She engages a lot of her colleagues and her company matches every single one of the gifts that her colleagues make to the foundation. So thinking about sort of even bigger picture, how we can maximize individual contributions through corporate matching. There's things like even more sort of complicated vehicles like distributions from life insurance policies and all that other stuff. That's really complicated, but I'm happy to talk to anybody who wants to sort of go to a different level with talking about different, more complicated mechanisms like donations of stock. And if that applies to your family, I'd be happy to talk to you about that.
Okay, we're going to dig back in. There's some really great science questions here too, and clinical questions. I'm going to do a quick one for Bianca. Somebody was asking about sending samples from overseas as well. Do you want to talk a little bit about that? And
it's logistically much more challenging for us to get things overseas to keep them stable. We need a really tight collaborator overseas, but if we have that, we have had success doing that. So I think talk to us about it. We're a little more targeted just because of the cost, the effort, and making sure that we can actually get the samples. So sometimes we'll prioritize different samples based on what the needs are of the biobank and the research community. But I really encourage you to reach out to us, and we're happy to work with you and see what we can do.
And the link to the registry email, to the natural history study email is on the ARRE Foundation website. Yeah.
Great.
Okay.
arrefoundation.org/registry.
arrefoundation.org/registry.
Need to update to natural history study.
Yeah. Okay, Stephanie, can you clarify whether the genes that are affected by ASXL have been isolated so you know exactly which ones to study?
Yes and no. So, I mean, it's in some cells, they are better described than others. So I described, and Bianca has emphasized that cells are changing all the time. And so that has, in order to try and understand what the ASXLs are doing, that's required researchers to hone in on specific cell types, which only represent a small snapshot of development in order to try and understand things. And yes, say a neuroprogenitor cell or a fibroblast, we do have a good idea of the genes involved, the genes impacted, and the genes being changed relative to ASXL1, 2, or 3, and even BAP1. But those are important, but I can't say I know this like for all cells across development. No.
I mean, I think along those lines too, part of the reason that it's important to have multiple model organisms, is we talked a little bit about the developmental process. And so different organisms can help us study different stages of development. So for example, we heard a talk when someone who's studying worms. You think, worms, how does that relate? But the worms are translucent and you can see through them, and you can watch the cells migrate and form sort of the early nervous system, right? So that's a really powerful tool. Chick embryos, for example, you can look into the eggs and watch the early heart and the early brain cells form and the early gut cells form. So we can get kind of an eye into some of these different systems. We talked a lot about frogs that have these really big eggs and they develop really quickly. And you can also watch them and they make a lot of them. So you can study those and really watch the brain develop with these ASXL changes in those different model organisms. So there's some really cool, powerful tools and neat ways that we are able to sort of study that phase that we can't access in humans as well. And then someone else asked me a great question too about what about my kids' cord blood, right? So those are historically stem cells that we think of as being able to turn into any different cell types in the body. But our technology has gotten so good that if you give me your skin cells, we can go into the lab and we can turn your skin cells back into brain cells, back into gut cells, into kidney cells, heart cells. And so there's some really cool, powerful things we can do with the model organisms, but then also with the samples and the cells from your loved ones. And those themselves can be really powerful ways for us to actually go backwards and study what these genetic changes were doing during those phases when we weren't able to see what was going on. And another good point, Amanda encouraged me to add this too. I mentioned back to sort of the treatment concept. We definitely need to figure out what these genes are doing and develop treatments that are specific to them. But there's a lot happening in the neurodevelopmental space right now that's a bit broader. So there is a lot that happens where there may be a different genetic change that's impacting how the neurons in the brain develop, but it has a similar effect, right? So if the neurons in the brain don't move where they're supposed to be, or they don't have the normal structure that they're supposed to have, why that happened may not matter as much if you're able to give a treatment that now helps those neurons get to where they're going to need to be, or helps those neurons look more typical. And so I told a story yesterday about Rett syndrome and how they have a treatment that they're using in their community. And that treatment itself is actually not specific to MECP2. It doesn't do anything related to how that gene works or doesn't work. It works more globally in helping the neurons in the brain develop more normally. So it's really important that we do this work, and we're super focused on how the ASXL genes work and how we can help them. But it's also true that there may be more global treatments that may be available to the rare disease communities that aren't specific to our condition, but can be useful. So I think that's an important point too.
And I'll just bring you back to the deep phenotyping and the research that's performed on you guys, because we don't see how those similarities exist between all the rare diseases without deep phenotyping.
Bianca, you mentioned that there are a lot of technologies available and a lot of new technologies available. And this is a question here. So if we have all these technologies and we can make all these changes, and this is probably also a question for you, Stephanie, what holds us back from using one of those technologies to just fix the bad gene?
I think I would say a big challenge is delivery. CRISPR was very recently rolled out with FDA approval for the most accessible cells, such as lung cells and, cystic fibrosis, disorders like that. And I think that if you have many cells that need to be changed, then that's a challenge. And I think there are gains being made in ways to harness CRISPR editing, DNA editing, so that it's really specific. But at this point, it's still really kind of leaky, and you have to have a lot of confidence that you're only going to change the thing you need to change, because any other change could be just as bad as what you are trying to fix. So those would be two challenges. I'm sure there are more.
No, I think that's a great point. And then another thing that I think, Stephanie, you and your lab have talked a lot about is there may be multiple functions, right, of these genes. So the idea is if we knock out the gene function, that may not be the only, we may cause another problem somewhere else, because somewhere else the gene does something else. And so if we have multiple functions of these genes and you just target one and you knock that out, the body has an incredible way of compensating and coming back with something else. So you really do have to understand the biology and what you're doing in various cell types and various times in development in order to safely roll out these technologies in the human body.
So I think it's really important one of the distinctions between Bianca's comment and mine, CRISPR can be used in such a way to make, specific changes that could correct and make a normal ASXL sequence, but it also can be, if deployed in the wrong way, it will just make more inactive genes, and that's a bigger problem. So it's important to know what people are proposing when they say CRISPR, because it can be interpreted in many different ways.
I think we maybe have time for one more, right? 9:30 is more. Okay. So I'm going to toss out one of the more in-depth science questions real quick, and then we can wrap it up, which is theoretically ubiquitination would then be what gives the range of pathogenicity or level of severity. Is that's the question? I'll toss that over to you.
It is possible. And while I described ubiquitination as a discrete one ubiquitin to one gene, it isn't that simple. And we actually have a lot of ubiquitin across the genome. And because of that, it's easy to just cause small changes in how genes are activated or turned on and turned off. And changing how it's turned on even by a day or two can, over time and the course of development, have really complex implications, just because these genes all need to be turning on and off quickly in the right combinations. And so in some cases, that may be one day, in other cases, it may be 2 days. So that could have a big impact on the severity of symptoms, even though the same mechanism is disturbed.
And this is a really hard question. And I know a lot of families have asked us, why when you look at the room and you put all these families together, how do we have such a spread? Why does it look so different? And this is a common challenge in genetics in general, in that we call it the phenotypic spectrum. So if your phenotype is the physical manifestations of a condition, and there's always a spectrum. It doesn't matter what genetic condition you're talking about, there's always a range of features. And there's lots of work that's been done generally to understand why that's the case, more broadly, sort of as a general genetics concept, and then more specifically into different conditions. And there's lots of factors. There's lots of disease modifiers. There's lots of other things that aren't the ASXL genes themselves that are happening in the body to sort of fix some certain processes and make adjustments. And we honestly really have no idea in most situations what it is that is kind of controlling the differences that we see across a condition like this. So this is a common, well-established sort of phenomenon that happens in all genetic conditions. And it's really complicated. So you can always have components of it, right? Like that differences in ubiquitination may be a piece of it, but it never is the whole story.
Yeah.
There's always lots of complexities that we just don't understand.
Agreed.
Thank you all. There's one last question in here. I want to wrap up, and I think I'm going to take this one and answer this one, which is how long until we have a treatment or a cure for any of the ASXL disorders? And I think from Amanda's perspective, my perspective, Laura, and the rest of the board here, the answer to that question is really how much engagement we get from the families, right? The more engagement we get from the families, the more you answer our calls when there are studies that need to be done, the more that we can do, small fundraisers and help fund some of these travel grants for these young researchers. It's been amazing what we have been able to do just over the last 3 years, over the last 5 years since Laura started this foundation, and you saw the growth of the research community in those photos that Amanda shared. We can accelerate the journey to these treatments and to any potential cures by engaging with the foundation and engaging with the scientists. And it's one of the reasons that we bring the scientists and the clinicians all together with the families every other year to help move that forward.